How can we see color? I would like to start off this presentation by asking and answering the question, how can we see color in light? We first must look at the structure of the eye and understand how light is interpreted or processed within our eyes and brain. We will only be focusing on certain parts of the eye that relate to this topic. Structure of the eye. Take a look at the picture of what the inside of the eye contains. The front of the eye, which is the cornea and the lens, is just responsible for taking in light and flipping what we see. The flipped image is then sent to the back of the eye, where it shines on the retina. The retina is a light-sensitive mass of thin layers that process the light. After it processes the light, it converts what we see into an electrical/neural signal that is sent to the brain via the optic nerve. It may seem as if the hyaloid canal is responsible for transferring light to the back of the eye because it looks connected. However, its purpose should not be confused with the retina, which covers the back of the eye. The purpose of the hyaloid canal is to adjust and stabilize the amount of vitreous liquid in the eye. Vocabulary. The retina is the back of the eye that is sensitive to light. Several layers of changing cells that exchange light/image into a neural signal that can be sent to the brain. Rods/cones are photosensitive cells, and the optic nerve is a signal that is sent through this nerve from the retina to the brain. Spatial acuity is how clear you can see and the sharpness of objects around you. Scotopic is relating to vision and dim light, and photopic is relating to vision and bright light. The fovea centralis is a small dimple in the middle of the retina. Quote, it is the center of the eye's sharpest vision and the location of most color perception. Rods and cones. Within our retina, we have two photoreceptors called rods and cones. Starting with a rod, they are scotopic, which means that they are most useful when we are in dim light. There are millions more of these in our eyes and cones and more sensitive. However, they are not sensitive to color, but rather to the amount of light there is. They have low spatial acuity, but are most useful for night vision. Moving on to cones, they are very different from rods. They are photopic, meaning they are most useful in bright light. Unlike rods, they have a high spatial acuity, meaning they are responsible for how clear and sharp everything seems. Also, they are found mostly in the fovea centralis, whereas rods are not. They are also sensitive to colors. Cones are a bit more special than rods because they are split up into three subtypes of color reception. The three types are red, which are long wavelengths, blue, short wavelengths, and green, medium wavelength, color-sensitive cones. Most of the green and red cones are located in the fovea centralis, but the blue cones are located outside of it. Despite the fact that the red and green cones highly outnumber the amount of blue cones, the blue cones have a very high sensitivity, so they all balance out. Fovea centralis. Now that we've established what exactly rods and cones are and how they play a part in how we see, it's time to elaborate on the fovea centralis. So where exactly is it? Is it the same as the retina? Not quite. It is actually more of a specific area in the middle of the retina, a small indentation. As you know, this area only contains cones, and it is where we have the highest acuity and sensitivity to color. Real-life examples/topic uses. This information is useful in many ways. One reason why knowing what our eye is made up of is so that we can understand the reasoning behind diseases related to sight, such as color blindness or cataracts, which is blurry vision. This knowledge can also help us figure out why some people may experience cloudy vision, strange flashes of light, or have dark spots in their vision. Knowing about the eye, particularly about the specifics of rods and cones, can help scientists understand more about the eye and vision. This information can help them develop medicines or surgeries that can improve the eyesight of people with vision problems.